I'd like to introduce Dr. Logan Kistler from the Museum of Natural History at the Smithsonian.
Can you hear me? Sure. All right. Well, thanks to everybody for being here, and thanks so much for Jim and Jana and the organizers for pulling this together. I'm really happy for the opportunity to come and be part of this conference. I'll be the first to tell you I'm not a Florida archaeologist, so I'm a little bit of a fish out of water, and in that way, I'm afraid I'm not going to talk to you a whole lot about Florida archaeology, but I wanted to tell the most Florida possible story that I could to come share some of my research with you. And I thought that should definitely involve these wonderful mastodon remains that are coming out of the wet sites in Florida. So what I'm going to try and do is link up our seasonally favorite vegetable with our favorite mega beast from the Florida wet sites and put together a story of the domestication of plants in the context of indigenous cultivators. So I'm not a Florida archaeologist. I did start in Kentucky archaeology. So that little pinpoint of light is me 10 or 12 years ago in the rotunda of Mammoth Cave, and it's difficult to ignore the origins of agriculture when they're sitting on the surface of the sediment untouched for the last 3,000 or 4,000 years. So this is when I started to get interested in the evolution of domestic plants. And this just by way of a little bit of background of how we get into the story that I'm going to talk to you about today. In sort of the Mammoth Cave, Kentucky archaeology context, I got interested in the evolution of the Chenopodium, the Iva annua, the sunflower, and the squash that come out of the Eastern archaeological complex. A lot of those plants that drove a lot of the societal changes or facilitate a lot of the societal changes in pre-colonial times and in pre-maize times. But I got interested in working with the genetics of plants rather than strictly with the archaeology of plants. And so that drove me into more lab-based and museum-based science. So I swapped one rotunda for another, and I'm now based at the Smithsonian, working primarily with genetics of plants. And that's where I'm going to focus and try and pull that together for you with the archaeology of squashes and pumpkins. So this is Cucurbita pepo, but this is the wild kind. It's the same species as all the zucchinis, many of the summer and winter squashes, and the jack-o'-lantern pumpkins that we're all going to be buying pretty soon. Cucurbita pepo is one of about 14 species in the genus. They're all New World. They're all from South, Central, and North America. This is an example called pepo subspecies ovifera var ozarcana. It's something from the Ozarks of Arkansas. And even though there's about 14 different species, this is a really diagnostic form of this plant. It's this long scrambling vine. It's got these little fruits, which are as hard as a softball and about as tasty as one, too. They're not really much of a food product, but this is a good example of this species. There's 5 different domesticated species of this group of plants. So the butternut squash is a different one. The cachaz is a different species that comes out of Central America and Mexico. So there's a lot of diversity, and there's a lot of sort of convergence in terms of domesticated forms of squashes emerging throughout the Americas from about 10,000 years ago up to the present. So these fruits, these pepo fruits, are not fit for humans, but they're fit for our friends here. These are classic megafaunal fruits. They don't dehisce, meaning they don't break open and disperse their seeds naturally. They have a very thick rind. You have to open these with a machete or with a hacksaw. They're a couple millimeters thick. And if you let a field just dry out, those will just sit on the surface and go nowhere. There's nothing that disperses these today in the Americas. But they have the characteristics of these megafaunal fruits, the really heavy sort of architecture of the fruit, and also, I'm missing a slide, I'm afraid, but they have one other characteristic, which is a chemical defense. So these squashes are inedibly bitter. They're incredibly bitter. They have a chemical compound called cucurbitacins. It's a family of compounds that's the most bitter naturally occurring substance known to humans. There's a phenomenon called toxic squash syndrome, where if sometimes you're growing a zucchini or even a cucumber in a different genus, and it gets infected by certain insects, or it gets attacked with certain stressors, or it gets cross-pollinated by a wild plant, that then domesticated plant, the zucchini or the melon or the cucumber, whatever, starts to express these bitter compounds. And you'll know it really fast because they're just terrible. And now, if you don't listen to your evolutionary signal and stop eating that bitter gourd, you're really going to regret it. Because these bitter compounds, I don't know why my Slides have just been wandering forward. Pardon me, I didn't even notice that was happening. These bitter compounds are toxic. That's what bitterness is in an evolutionary context. It's telling us this is a thing you don't want to be eating, spit it out, and you get to stay in the gene pool. So chemical defenses and this physical overbuiltness are both signals for megafaunal fruits. But in the case of cucurbitas, we have more evidence than just it looks like a megafaunal fruit, and it looks like fruits that rhinoceroses in Asia and African elephants are eating today. Because, of course, in Floridian sites, we have the digesta of mastodons. At the Page-Ladson site, more than 12,000 years old. At the Latvis-Simpson site, more than 30,000 years old. And sure enough, this is before humans could have been influencing the evolution of these plants, we find squash seeds. And so a lot of this, the identification of the squash seeds and the characterization of those was work from my former PhD advisor, Lee Newsom. And it was conversations with her that led me to thinking about how mastodons and humans are sort of two sides of an evolutionary picture as concerns the evolution of squashes. So we have this direct evidence that mastodons were dispersing these terrible little tough bitter fruits that are unattractive to humans. That puts them, because all the mastodons have disappeared, in the category of ecological anachronisms. And what we're speaking about there is plant species who are left sort of high and dry, out of time and place, with a co-evolved mutualistic partner. So these things are evolved to be dispersed by mastodons. Mastodons disappear from the landscape. Therefore, these are ecologically anachronistic. They're missing something about their fundamental ecology. This is an idea first introduced in the early 80s by Jansen and Martin. And they identify several species, things like the Osage Oranges, honey locusts, these sort of overbuilt species. And then this was really nicely expanded on into a full book by Connie Barlow, The Ghost of Evolution. We know of a lot of other examples of these. These ecological anachronisms, once you start to get in that mindset of looking for dispersal mechanisms that might not be on the landscape now, there are actually quite a bit of them. Avocados are thought to be Gomphotherium fruits because that big old seed is not really able to pass through the digestive tract of much else. On the inside of that seed are some mildly toxic alkaloids. Cacao. There's this lovely sweet pulp on the outside of every seed, but in a massive, tough, non-dehiscing fruit, and inside of that are some slightly toxic seeds. Remember, caffeine and theobromine are defenses against herbivory. We've just, as humans, taken a slightly different perspective on that. Osage oranges, they've got that latex on the inside. They're really not fit for much, but it is thought that the prehistoric horses could have been interested in those. And these massive honey locust thorns, this is so excessive given any mammals on the landscape in eastern North America today. But if you start to consider the sort of the mass and the magnitude of what was wandering around before, it starts to make sense that you would need to overbuild your defenses. And so the sort of the commonalities between these ecological anachronisms are the physical defenses and chemical defenses, and our squashes certainly have both of those, the physical defenses and the bitter compounds that are the chemical. So one thing we see is that we have cucurbita in the paleontological and archaeological sites in Florida in the late Pleistocene. We have it into the Holocene in the sort of the coastal plain in Georgia and South Carolina. But then, pardon me, we don't have any wild cucurbita populations to speak of in a lot of these areas now. It seems like it was much more connected. It was much more robust population several thousand years ago, but we can sort of see it pulling back into what's now more or less refugial relict pockets. And it's not real easy to find this plant in the eastern US now. And so there's a couple reasons we think that could be the case. One is they're losing their dispersers. Those large herbivores are all disappearing from the landscape. We don't have any herbivores more than 1,000 kilograms, or especially not these giant sort of generalist browsers that you had in the Pleistocene. And along with the direct loss of the herbivore, or of the dispersers, you have the loss of the effect of that massive herbivory on the landscape. And you have the loss of the weedy niche created by those large herbivores. If you look at where savanna elephants and forest elephants are, they are shredding the landscape. They're creating all of this mosaic-like, niche-diverse habitat, and that's perfect for a weedy plant that loves to colonize disturbed areas. And that's exactly what cucurbita is. When you find it out west, in the southwest where there's another species, it tends to be on the road edges, tends to be on the fence rows, it's in disturbed habitat. It's a weedy plant, like many of our domestic plants. And so as these massive herbivores are disappearing, they're robbing both the dispersal strategy of the plant and the habitat that they have created in which these things have thrived for probably millions of years. And so they're disappearing in domestication, but they're disappearing in the wild. In the same time period, from 10,000 years ago to now, we've got a very different story happening in the context of humans. So humans are selecting on these things, they're embracing them in a lot of sometimes fairly extreme ways, but they're creating out of this bitter, inedible fruit something that is really much more familiar, much more diverse in phenotypic terms, and now completely ubiquitous. So while it's declining in the wild, it's gaining a ton of traction in domestication, where we come to know it today. At the beginning, it's kind of unclear how people started using it because it's toxic and because it's bitter, and it could have been a utilitarian species, like a bottle gourd. So bottle gourds, of course, were much more ubiquitous as containers. They're eaten in some places, but that's not most of their usage. And there are cases, I believe this one's from Key Marco, someone can correct me on that, where a cucurbita is used as a fishnet float. The seeds are a little less bitter, so there's some ways in. This is kind of a black box of the evolution of the thing so far. So hopefully we can get into that someday. But it seems like at the beginning, there's a good chance we were looking at this as a utilitarian species to sort of get humans more familiar with it. This is a slide borrowed from Lee. The evolution of cucurbita and the archaeological record of cucurbita, of course, Florida is a real nexus for these sites, and the wet sites in Florida. Not only the paleontological ones, but the archaeological ones have been really key for understanding some of the early dynamics of these species. So the way we started thinking about this is we have this plant that's declining in the wild, but it's gaining ground in domestication. So we can start to think of domestication as sort of an alternative evolutionary trajectory. It's plants sort of changing their tack in order to survive in a changing landscape. So the questions are, how widespread is domestication? Where are these sort of trajectories of domestication originating? And if you have the disappearance of these wild stands, is that affecting how these plants are evolving by changing the amount of diversity that's available for breeders, for indigenous farmers to be working with and creating new varieties? And does that provide a sort of reproductive isolation that helps indigenous farmers breed new varieties? So these are the questions that we wanted to get into. So we collected a lot of land races. We went to the USDA. We got about 70 modern types. And we collected about a dozen archaeological samples. And I won't get into the technical elements of this, but we were able to sequence a significant portion of DNA from each of these individuals. And by sequencing the DNA, we can build family relationships. We can compare the similarity and the difference of the sequences of the base pairs. And on that basis, we can say what's more related to what and what derived from what. And by putting that on a landscape, we can start to understand a little more about those questions. We do that in a specialized clean lab. I'll just show you kind of what it looks like. We irradiate everything with UV. We clean everything with bleach. This is all just to make sure we're working in clean conditions because it's easy to contaminate these experiments, but I won't dwell on that. I want to show you a few results from these experiments. So this is a diagram showing the relatedness between the different individuals. And the only way you need to think about reading this is the amount of horizontal distance between two of these tips that you have to travel is correlated to the amount of evolutionary distance between them, how related they are. So if you look at this Cucurbita okeechobeensis here, it's a very short horizontal distance to this Cucurbita lundeliana. If instead you look at the maxima here, it's quite a significant distance to the ecuadorensis here. So things that are closer together on the diagram are closer together in evolutionary terms. And then what we're showing here is some of the archaeological sites and which species are present and the relatedness between those species. So I'll highlight a few things on this. The first is this subspecies ovifera. And this has been sort of argued about for 40 years or so, whether eastern US is an independent, a truly independent emergent site of the domestication of squashes. And we found really clearly that it is. The ancient materials from that area exactly match the modern materials, exactly match land races and cultivars within that subspecies. The next thing I want to highlight is to have a look at this Cucurbita pepo subspecies pepo. That's your zucchinis, your jack-o'-lantern pumpkins, some of your really more recognizable ones that we grow in our gardens today. And look at this, the prehistoric span of that species. Now, that's not a species that we have in the present day in the wild. That one seems to have completely disappeared. People have gone looking, and it seems like during that sort of ecological pullback, it completely was extirpated in its wild form. But in prehistory, it's being grown in the Colorado Plateau. It's being grown almost as far south as the Yucatan. It's got this huge spread, which leads a person to ask, is this because it has a huge ancient range, or people are really moving this plant around pretty early on? And given the radiocarbon dates we've got off of these, it has to be one of those two. So we see these really sort of widespread episodes of domestication. The third item I want to point you toward is this little guy, Cucurbita pepo subspecies fraterna. That's a wild gourd. As far as we know, it has nothing to do with domestication. It has nothing genetically to do with any modern domestic. We have looked. But we have that from an archaeological site in Tamaulipas. We found that species, and it looks to be in a slightly domesticated form. It's got these furrows, it's got a thickened rind. And so what it's showing is, given the diversity on the landscape now, is a small subset of the diversity that was in crop fields thousands of years ago. And there are entire species and subspecies that have disappeared for one reason or other from crop fields. And so we should be thinking about domestication in this much broader ecological sense rather than saying the species we know now is the same as the species we knew from 10,000 years ago. It might not quite always be that simple. Last thing I want to show you from this is have a look at these three samples here. Cucurbita okeechobeensis is native to Florida. It's endemic to Florida. I think it's only known from Lake Okeechobee and St. John's River in recent times. Okeechobeensis subspecies martinesii is actually across the Gulf there in Mexico, as well as Lindeliana. I'm going to explode this graph out into a much more technical, much less nice-looking version. But what I'm showing you is pay attention to how related these are to one another. We're going to look at the exact same graph, just blown up. That's those three tips. They're incredibly genetically restricted. They're almost identical. For comparison, this is the Cucurbita pepo group. That's all your zucchinis and your jack-o'-lantern pumpkins. Compared with those, these are incredibly genetically related, and yet they come from across the Gulf of Mexico from one another. So what this speaks to is the fact that these little pockets of endemic things that we see nowadays are actually the remnants of a much more connected population that not that long ago, maybe with the help of giant herbivores, was a connected, dynamic, communicating population, possibly with the help of human cultivators. And so what we see is domestication was widespread with these failed lineages, these experimentations and these fits and starts. We see that there was prolific human-mediated dispersal of some varieties, or their home ranges were just huge and domestication was happening in several different places, which we already know was happening in other groups. And we have recent fractionation of these closely related wild types, which tells us something about the ecology of the plant pretty recently. So domestication was widespread during natural declines in the wild. So let's return to those natural declines in the wild and figure out a little bit more about why these things were so evolutionarily compelled to pull back and to, in some cases, disappear completely, particularly in the tropics. So let's return to the bitterness and the dispersal ecology of these plants. We know that mastodons were chewing these things up and dispersing the seeds. But we've already stipulated that these things will make you incredibly sick if you eat them. So we wonder if there's something about the relationship between the fact that these animals are huge and their ability to eat them, not taste or be bothered by the bitterness, and not be poisoned by them in the process. So we wonder if there's some fundamental difference between very large animals and very small ones in terms of their ability to interact with these toxins and this bitterness in the environment. And I will get back to domestication in a moment, but bear with me. So we looked at every mammal that we could. So this is all based on published data. So we looked at all the mammals which genome sequences have been produced for. And what we could do is look in them for a particular gene family. We call them the taster genes. They're responsible for how well and how broadly we taste bitter compounds. The more copies of the taster genes you have, the better you are at tasting and detecting bitter compounds in the environment. Humans have 24 or 25 copies of these genes. The difference between the people who have 24 and the people who have 25 is the difference between whether or not you're a big fan of things like black coffee and Brussels sprouts. You can tell phenotypic effects of these, and they're incredibly variable throughout the animal kingdom. So we looked at all the mammals we could, and what we found was this huge association where body size is incredibly predictive of the ability of these things to taste bitter compounds. So the really big animals, the manatees, the pandas, the elephants, they don't have too many copies. They still have to taste some toxins, but not so much. And then the shrews and the mice, they have up to 46 copies, down to about 6 in the manatees. So body size seems to be related to the ability of these animals to detect bitter compounds, including the ones in our squashes. Not only that, but if you start to compartmentalize this further, you start to think about the sort of the feeding ecology of these species. Some are sampling a lot of things in the environment. They're very much generalists. Others are specialists. Specialists don't need to taste all the bitter compounds because they're only sampling 1 or 2 things. Think about your panda bears. They're only eating bamboo. It doesn't matter if the next plant over is poisonous. And so what we found is the more specialized a species is, the less able it was to taste bitter compounds. And we could actually explain like half of the variation in this bitterness with just these 2 traits, just the size of the animal and its dietary preferences. So what does this mean for domestication, and what does it mean for cucurbita, given what we know from Floridian archaeology and paleontology about mastodons dispersing cucurbita seeds? What it means is that because all of the really big animals, the really big herbivores have disappeared from the landscape, all of the animals capable of tasting the nasty chemicals in the cucurbita have also disappeared from the landscape. Anything that's left is much too small to be able to tolerate those bitter compounds. So there are no dispersal candidates left except for one. Because as the Holocene changes, there's the lack of the disturbed habitat, and there's lack of the dispersers. The other big change is that humans are starting to really affect the landscape at about the same time. And so humans provide the ideal alternative disturbed habitat, and they are willing dispersers. So humans can provide sort of a surrogate dispersal mutualist in this case if the gourds are willing to make a little bit of an evolutionary concession. Because if they still taste terrible, they're not going to be great partners with humans, and if you can't get through the rind or eat it without poisoning yourself, that's not going to work either. And so the evolution of the sweet flesh, the softness, the well-behaved plants that we know and love today is a result of these things cozying up to humans after their natural partners disappear. I want to briefly touch on this issue of reproductive isolation. So every domestication scenario, you have to at some point cut off gene flow from the wild. You have to isolate your domestic population from the wild, or you're just going to keep breeding wild traits back in. So we know, for example, in the cereals and in sunflower, we know that the cereals and maize became much more robust after they were carried away from the domestication center. That's when they really took off. That's where we can see morphologically that the archaeology gets really interesting with these plants. Sunflowers have a similar thing, but they have tons of gene flow. They can tolerate a lot more. And this is not restricted to plants. Of course, we've done very strange things with our domestic animals. But the point is that in cucurbitae, this toxicity means it absolutely must be isolated from its wild counterparts if it's to be used for food. If you look in the Extension Agency guidelines, like in the Southwest where there's wild ones growing, they will recommend rather extreme techniques of making sure you don't get cross-pollination if you're going to save your seed, because you'll end up with inedible crops. There's also apocryphal stories of one neighbor who didn't like his other neighbor, so he planted wild plants in his own garden to sort of carpet bomb the whole area with pollen from the wild plants. It must be isolated for food. So when the wild types disappear, what you have is this reproductive isolation is happening right in place. The wild stuff is just dropping away. So that's one barrier to domestication that isn't a problem because of this changing ecology. And to extend that, what we have to ask is if the wild ones had thrived on, if mastodons were still here, which would be outstanding, if they were still here, and they were still dispersing these. Would we have edible squash today? Would we have ever gotten to that point of reproductive isolation? I don't know the answer, but I like to think about it. I'm going to leave you with just one little bit of unpublished data. We're looking at a much more nuanced part of the genome from all of these samples now, and what we're seeing is the domestic type is more genetically diverse than the wild type. We've seen declines in wild type diversity. This is the exact opposite of what you expect. Because usually you have a wild population and human cultivators start to pick and choose, and they start to cull away the variation that's there, and you end up with a subset. What we have here is that humans are propping up the diversity as the wild plants are disappearing. Thank you so much for your time.